All right, this is our experiment, salty pea plants.
Okay, so our experimental question was how does watering pea plants with water containing sodium affect them and contribute to their growth?
Our abstract, so the specific anatomy of the Pisum sativum, which is the pea plants we did, starts in the roots, goes up to a stem, then to the pedicel, which is like a stem, into the flower of the plant, which contains the banner and the wings, which protect the ovaries. The ovaries are on the flower stem, which contain nice which both contain stigma, and that's what helps the plant fertilize itself and reproduce. The pea plants absorb water through their roots through osmosis. The water then moves from cell to cell inside the root cortex. Then it enters the xylem vessels, which distribute the water and its nutrients throughout the different cells of the plant.
Okay, so the effects of sodium we saw an experiment on the National Library of Medicine. They did a similar experiment to us, and they concluded that the higher salt index contributed to more salt stress, decreasing plant growth. So the way the plant affects the growth is it causes osmotic stress and ion toxicity due to the amount of sodium present. So this disrupts the cycle and causes the nutrients to not be transported throughout the cell or the plant.
So an overview of our experiment, it tests how sodium levels in water affects the growth of pea plants. There are three different groups, one control which received regular water, one group that received 100 milligrams of salt dissolved into the water, and a group that received 200 milligrams of salt. The plants were each watered with 1 tablespoon or about 15 milliliters of water, and they're watered every other day. We measured the growth of the plant from the soil level to the highest point.
Okay, so the materials we used nine pea plant bags. We used one bag of organic potting soil. We used nine separate containers, 4 inches in diameter each. We used sea salt, we used water, we used a scale, and we used a container capable of measuring out 1 tablespoon, as well as a semi-large container. And then that's Evan with his materials.
The procedure is first you fill a semi-large container with water and place the 9 pea plants inside of it. You soak the pea plants for about 16 hours to decrease the germination time. After soaking, you fill each container with about 3/4 full of potting soil and plant each seed into the center of each container about half an inch deep. Then you place the containers outside in an area where they all receive the same amount of sunlight. Water the plants every other day for 2 weeks. For the groups that require salt, you first measure out the salt on the scale and then you put the salt into the water you're using, mix it up so it dissolves properly, and water your plants. Repeat this process for each plant that needs salt water. When you measure the plants, measure them at the same time you water them every other day for 2 weeks. Measure them in centimeters and round the data to increments of 0.5 centimeters.
Okay, so the pictures we can see on the left is the pea plants like in the soaking in the water for the 16 hours. The middle shows the group labeled C for control, the 100 milligrams and the 200, the three groups of three. And then we see the scale on the right, which excludes the weight of the bowl in measuring the salt.
Our hypothesis is if the sodium levels in the water being used to water the pea plants are increased, then the growth of the pea plants will decrease.
Okay, this shows the graph, which is labeled in centimeters. So the control group, which is the light blue, starts at 7, where we see the first growth, and then from day 9 and on, it's a constant rapid increase. Whereas for the 100 milligram of salt group, it starts at day 9. When it hits day 11, it doesn't grow at all, remains constant for the rest of the experiment. And then for the 200 milligram group, it does not grow at all throughout the entirety of the experiment.
Our data tables are focusing on each group's individual plant. As you can see in the control group, it sprouts about day 7 and then rapidly grows. In the 100 milligram group, sprouts also around day 7, but does not grow only a little bit. And then the third plant doesn't sprout at all. 200 milligram group, no plant sprouted. Here are our pictures. These two in the top left and bottom left are of me and my partner, but we could not get a good angle of the plants when taking a FaceTime photo, so we didn't include them in these pictures. We just put them to show that we did the work together. This is on day 1, and it branches out of the plants, branches out into the control group first, where you can see um, it's sprouting and then growing quite rapidly as you get to day 11 and day 13. The 100 milligram group sprouts about the same time as the control group you can see here, but it doesn't grow very much at all throughout the rest of the experiment. And the 200 milligram group does not sprout at all, as you can see here.
Okay, so our analysis is that the data we collected supports our hypothesis that an increase in salt and water used to water pea plants causes a decrease in growth. Our data is used as evidence as our control group that was watered with water that didn't contain any salt just grew the most out of any plant. And as you add more salt to the water, the they like the it just decreases and doesn't sprout. So the growth is affected.
Um, Go ahead.
Some problems we faced were the salt measurement. So we couldn't find a scale that measured down to 0.01 centimeters at a reasonable price, which this basically means their measurements were off by about 5 milligrams. And then one of the days it rained, but Evan was able to bring in after 6 hours, but all of the plants received the same rain, so it shouldn't have altered the experiment. And if we were to redo the experiment, we would have used a more exact scale, even though it would have been more expensive, to make sure the measurements in the salt water were more accurate.
In conclusion, our experiment succeeded in accurately testing our hypothesis by providing evidence to help answer our scientific question. The experiment went relatively smoothly, and the process is very repeatable. Along the way, we learned how pea plants, their anatomy, how they absorb their water, and how sodium can affect their growth. We also learned how to set up, carry out, and analyze a scientific experiment.
So a world connection would be so we can see that in arid air or very hot areas, there would be more like saline in the soil, which means there'd be more salt. So this can lead to large scale food shortages as if we're not mindful in where we place our food and where we put it, it can cause like major like downfall in terms of like agricultural growth. So this means we have to be put them in tropical areas so they can survive and thrive.
And here's our works cited. And perhaps the best thing we learned, or not the best, the most important thing we learned was not from this experiment, but from the first one we did where we lied and we failed on it, and will talk
Yeah,
about that.
so basically, so how we did it. So our experiment, our whole idea was how harming bacteria affects plant growth. So we lied about the bacteria we used. We used something that we did not use, but we learned to come clean and we learned something that was more important in this class and something that we can apply later in our lives, which is basically that like sometimes even though it's hard, even though it's not easy for me and Evan to go in and admit our mistake, we showed integrity of like what a true man would do. And we would when we came in, we admitted what we did wrong. So
And thankfully, Mr. Heinen blessed us with a second opportunity to do this experiment, and we didn't take it for granted. We learned that it was through the hard work, dedication, and showing up when even you didn't feel like it. That's what helped us get through this experiment and make it better than our first one by not taking the easy way out, which is what we did in the first experiment that led us to failure and harmed us, set us back. By working hard on this experiment, it led us to success, and we've grown a lot from that.
yeah, so me and Evan were super apologetic for what we did the first time, and we're ashamed that it happened. But we're me and him were both super glad we were able to learn from what we did wrong, and we were able to change as human beings and be able to learn how to make mistakes and grow from them. And that's what hopefully we can both take later in life, and some like a big lesson we can learn after. So
All right, that's our experiment. Thank you, Mr. Heinen.
thank you.